Okay, so antibiotics, they're used widely in medicine, and so here are a couple different antibiotics that are used. They've been used since the early 1930s, and throughout the years, more and more have been discovered and implemented in medicine. However, throughout time with the use, there's been resistance that has developed. So for example, we can look at oh, this is off, that's why that didn't work. So we have penicillin here, and so it was deployed around 1941, and shortly after, in about 1946, resistance was already observed. So some of these antibiotics, it takes a little bit longer for antibiotics resistance to be observed, but for all of these that are shown, resistance has eventually been observed after implementation in medicine. Now, compounding that is the fact that there's a decrease in antibiotic discovery throughout time. So in the 1980s to 1984, there were about 19 new antibiotic drug approvals, and now in 2010 to 2012, there was only one. So not only are we increasing the amount of resistance we have, but we're having less antibiotics discovered that can account for that increase in resistance and the inability to use some of those antibiotics to treat the antibiotic-resistant drugs, or antibiotic-resistant microbes. Now, with those, there are estimated to be about 10 million deaths attributed to antibiotic resistance by the year 2050. So that's every single year, 10 million people are projected to die from this. It's a pretty high number. It's not something small to scoff at. So this is clearly a big issue that, if we don't do anything about it now, could become worse and worse throughout time with increased resistance. Now, how does antibiotic resistance happen? So this is a really kind of simplistic view of it, but I'm not going super in depth into how it occurs, so this general understanding is good enough. We have our microbes over on the left-hand side. We have some that are drug resistant and some that are not. Then we introduce the antibiotics. The patient takes the antibiotics. They kill the non-antibiotic resistant bacteria, as well as all the good bacteria that are present in the body. But the antibiotic resistant bacteria are left. And so they're allowed to grow and take over and multiply, and we end up with a lot more antibiotic resistant bacteria. In addition to this, there's also, once there's an antibiotic resistance bacteria, they can transfer their resistance to another bacteria through horizontal gene transfer. And this is all compounded and increased by concentrations under the minimal inhibitory concentration. So the minimal inhibitory concentration is the concentration in which visible growth of the bacteria is, inhibition of the visible growth of bacteria is, sorry, I'm trying to go off of, can I pause for a second and get my laptop for it? Because I have notes on my presenter view. And just be 1 second. Okay, sorry about that. I just have some notes that I need. So the minimal inhibitory concentration is the lower concentration of a drug that, under established conditions, inhibits the visible growth of a target bacterial population. And often in patients and in the environment, these antibiotics are present under that minimal inhibitory concentration. So being present under this concentration adds selective pressure to the bacteria to increase mutations for the antibiotic resistance, as well as it pressures them to share those genes through methods like horizontal gene transfer to other bacteria. So it has been found to increase resistance. Now, antibiotics into the environment, as I briefly mentioned, that can be a source of pressure to increase mutations and increase antibiotic resistance. And there's a lot of different ways in which antibiotics can come into the environment. We're going to start here at the manufacturing process. So antibiotics are manufactured, and then that has a waste stream that goes to a wastewater treatment plant. From there, the antibiotics also, they can be administered by a doctor to a patient, and then that patient will excrete antibiotic out, and that excretion will go to a wastewater treatment plant. Now, part of the reason why this is so important to consider is that humans excrete about 75% of the active form of the antibiotic into the environment. So we're really only metabolizing about 25%. And this depends on the particular antibiotic, but on average is about 75%. With the wastewater treatment plant, that treats the wastewater stream and then discharges it into a receiving water. So as much as, sometimes the antibiotics can be degraded partially through this treatment process, normally not all of the antibiotics are degraded fully and antibiotics are discharged into that receiving water. There's also aquaculture treatment with antibiotics. So aquaculture is like the raising of and breeding of fish. And as much as we would like those to be their own separate entities from the water, there is some mixing between waters, between the farms and the receiving waters. We also have inappropriate disposal of used containers and unused medicine. So if people don't finish their whole antibiotic course, they might dispose of it improperly, and that can end up either in our receiving or in our wastewater treatment plants if people flush it down the toilet, or it can go into our soil if they're tossing that in the trash and it goes to a landfill. From there, we have some animals. Just like humans, animals do not metabolize all of the antibiotic, and they're, again, about 75% of the active form they excrete. So with companion animals, they might give your dog or cat an antibiotic for an infection, or they might have some sort of surgery, and then they go do their business outside, and that interacts with the environment. As well as for livestock, their manure is often used for fertilizer, and that has interactions with the soil. Now, any antibiotics that are in the soil can be washed off by rainwater, they can sorb to the soil, and there can be interactions between water and the soil. To give a brief overview before I go into each of those specific categories a little bit further in depth, there are different classes of antibiotics. Don't worry about the majority of this. I just want to give you a general idea that there are various classes of antibiotics, and within these classes, there are various different antibiotics. So the ones that I'll be focusing on are the beta-lactams, the fluoroquinolones. They just have a fluorine versus the quinolones don't necessarily. Sulfonamides, tetracyclines, and macrolides. Not super important, but just familiarizing you with the names of these as I speak about them in further detail coming up. So the antibiotic sources, we're going to start with wastewater treatment plants. And as I mentioned before, humans excrete 30 to 90% of an antibiotic in its unchanged active form. That's the general range that includes many different antibiotics instead of the 75% average. And so again, that just depends on the specific chemical properties of the antibiotic. Antibiotics can also be, when you take them, they can also be changed into metabolites, so they can be partially degraded. But through the wastewater treatment plant process, they can also transform back into their active form. So even if you're only excreting, say, 30% of the active form and you have 60%, say, metabolites, that 60% can reform, depending on the antibiotic, back into the active form of the antibiotic. And so you can have a lot greater percentage coming out into this waste stream. There can be up to 100% removal from wastewater, but this really depends on the specific compound because it's such a diverse set of compounds with diverse structures. They react differently to treatment. It also depends on the treatment type. Two common types are chlorination or ultraviolet treatment. And chlorination has been found to perform better for all of the compounds except for ciprofloxacin, but that's all of the compounds that they tested. They only tested 7 compounds. So it depends on which compounds you're talking about, which treatment is better for the antibiotics. Antibiotics can also sorb onto sewage sludge, so even if it says it's 100% removal from wastewater, that doesn't mean it's 100% degraded. it can be that 100% sorbed onto that sewage sludge, and this is particularly important for tetracyclines and fluoroquinolones, which are two of those classes of antibiotics. Overall, it's a challenge. You have this challenge of treating a class of compounds with a diverse set of properties and trying to have a one-stream way of doing it, but it's not a one-size-fits-all. They are diverse and they might need to require a couple different steps. Moving on to another big source are hospitals. These are a big source because it's a high concentration of antibiotics in a small space, as well as a high intensity of antibiotic treatment. So often in these hospitals, there's a lot more people taking these antibiotics than in the normal population because of surgeries, because of different infections they're in there for. And then you often have them at a higher dosage or for longer periods of time. But despite this, I found in Europe only 5 to 10% of the total exposure to antibiotics is actually in hospitals. So it's a small percentage of the total exposure to antibiotics. This effluent is often treated by the hospital or by a wastewater treatment plant. So that's an important thing to consider, because if it's not treated by the hospital and it's treated by a normal municipal wastewater treatment plant, and that municipal wastewater treatment plant is not used to having that higher load of antibiotics and other drugs, it might not be equipped to be able to handle degrading and getting rid of these contaminants. Going into agricultural animals, this is a huge source of antibiotics or usage of antibiotics, because greater than or equal to 70% of antibiotics are administered to livestock. So that's the vast majority. And they're used to treat diseases, promote growth, and prevent disease. Now, there has been a recent change in the US due to the FDA, and they came out with this new law that says antibiotics that are important for human medicine can no longer be used for growth promotion or feed efficiency. And this came out in 2017. So due to that new law, antibiotic use decreased by 33% from 2016 to 2017. So it's quite a big change just by this one law. But it's just one step in trying to decrease our antibiotic use in order to decrease our impact on the environment and decrease resistance ultimately. With that, cattle consume about 42% of the antibiotics, and swine take up the other majority at 36%, and tetracyclines, one of the classes of compounds, are the most heavily consumed in agricultural animals. The last source I'm going to talk about is aquaculture. So aquaculture is the breeding, raising, and harvesting of fish, shellfish, and aquatic plants. And as I mentioned before, as much as we would like to believe that these are separate entities from the waters they're nearby, they're not. This Benbrook et al. In 2002 found that within 50 meters of these aquaculture farms, 74 to 100% of the wild fish contained antibiotics in their muscle. So these antibiotics that they're using to treat the fish that they are raising are ending up in the environment, and that's affecting those fish. It's getting out there, and so that's certainly an issue. And so also the regulations vary between countries for these antibiotics, and this is also the case in all of the other sources that I mentioned before, but I have a particularly useful example here to highlight. So we have the antibiotic on the left-hand side, and then different countries. So in Chile, you can use all of these antibiotics, whereas in the US, you can only use 2 of them, and in Norway, you can only use 3 of them. So even though the US might have more stringent laws about these antibiotics, it might not be the same case for, say, Chile. And Europe has more stringent regulations on antibiotics than the US does, and this is a problem because it's a global issue. As much as we would like it to just, if we tackle this antibiotic issue, all antibiotic resistance will go away in the US. That's not the case due to how global we are nowadays. So with that, just a quick recap of what I've discussed. The major points: agricultural animals use the vast majority of antibiotics. Wastewater treatment plants are big point sources of antibiotics, so they have a very specific outlet of where they come out. And then antibiotic release into the environment can lead to antibiotic resistance development. With that, I'm going to move on to my specific project. which is entitled Defining Minnesota's Antibiotic Footprint. And the purpose of this project is to go around Minnesota and try and look at the antibiotic concentrations, where they are, what other factors are around them, and also we're collaborating with another group that is looking at the antibiotic resistance genes that are present. So we're trying to come up with, why are antibiotics which antibiotics resistance genes are where, why are they there, which antibiotics are there, is there any correlation, is there anything we can parse out of that? I will only be talking about the antibiotic side right now. We're still working on getting all of the data and putting it together and analyzing everything. So, last summer, I went around around Minnesota to all of these different sites. And we chose these sites because they're either agriculturally impacted, meaning specifically agricultural animal impacted, or they're wastewater treatment plant impacted. So they're about half and half for each of those. And they were largely also determined by where we could access them. As much as we would love to be right next to a farm in a lake, it's not always possible because it's private land and there's not necessarily a public boat landing that we can go on. And to do a little explanation of how the sampling occurred, within each of these dots, there are multiple points. Some of them you can sort of see, but a lot of them were too zoomed out to see the difference. But for example, for the wastewater treatment plant impacted, we have, so the water is flowing this way. We have an upstream of the wastewater treatment plant effluent. We have one at the point of the discharge, and then we have anywhere from 1 to 3 downstream of the discharge, depending on the size of the lake, depending on where we could access boat landings and such. With the animals, we generally did 2 to 3 sampling points along the water bodies to try and get some sort of gradient, see where we could compare what sources are around it, and maybe kind of parse out some of those details. The way the antibiotics I looked at, kind of showed up weird. Anyways, there's 25 different antibiotics. We chose 5 of the major classes, and then also a non-categorized or others class that exists. And these were chosen because they're used most commonly, as well as some are used exclusively in humans, some are used exclusively in animals, and some are used in both, as well as some are natural and some are not. So it allows us a great array of, is it natural? Was it used in humans? Trying to parse out these differences and why things are where they are in the environment. So for sampling, we went out in a canoe into the middle of a lake or a river, and then we took a jug of a 1 gallon jug and sampled the water out of there. And then once we got back to the lab, I split each sample up into 3 separate samples. 2 of those samples I just left the same, kept them the same, and then 1 of them I spiked with antibiotics. This is to account for the different particulates in the water sample, as well as specifically like dissolved organic matter, because that can affect how the antibiotics are extracted, how efficiently they're extracted and analyzed for. So we can account for that difference in each sample. There's also a method link to account for contamination. From there, I adjusted them to pH 3 in order to preserve the sample, prevent any reactions from happening. We added EDTA that complexes metals, and a couple of these antibiotics also complex metals, and so we're taking the metals from the antibiotics and complexing them to EDTA so we can actually analyze them. And then we add surrogates, and these are just so we can make sure the sample processing goes all right and we can account for that. I'm not going to get into too much detail about that. Then I go into this thing called SPE, or solid phase extraction. And this has a sorbent bed right here. And that captures, we send our sample, a whole 1 liter sample through this cartridge, and the sorbent bed captures all of those particulates, and which include the antibiotics. Then we wash it. I wash it with a water methanol mixture, and that takes off some of those particulates. It leaves our antibiotics. And then finally, we put through methanol, and that washes completely off our antibiotics. We collect that eluent. A couple more steps are done, and then we can analyze it on a HPLC or liquid chromatography tandem mass spec. Pretty much it allows us to be able to differentiate these antibiotics because so many of them are so similar to each other. If we didn't use this, we wouldn't be able to tell the difference between them. Now I'm going to go into some of my results. We're going to start with this wastewater treatment plant effluent. I'm going to take a second to explain this slide and how everything works because the next like 9 or 10 slides are all a similar layout. So in the bottom left-hand corner, we have a map of Minnesota. It shows you, the star shows you approximately where in Minnesota that sample was taken. Over here, we have all of the antibiotics that I tested for. The ones that are grayed out are ones that were not present in any of these, and the ones that are not grayed out were present in at least one sample. The size of the circle represents the total sum of the antibiotics. So, for example, this has 2,326 nanograms per liter of total antibiotics. And so that's going to be larger than one that only has 379 nanograms per liter of antibiotics. And then the asterisks, don't worry about it too much. It's more of a technicality thing, but it just signifies the value that's between the limit of detection and limit of quantification. Does that make sense? Are there any questions on that? Okay. With this one also, these different colors represent the different sewer sheds that go to the wastewater treatment plants. But yeah, that's the general gist of these maps that are going to be coming up. So let me know if you have any questions on them. So with the wastewater treatment plant effluent, this is all from the Metropolitan Council, all the wastewater treatment plants that are owned and operated by them. They have since acquired a new one, but I have not tested that yet. And this one, Seneca, is missing because the sample I spiked the samples with antibiotics, and this one the matrix was so funky or weird that it just didn't work. And so I had to throw that one out in order to make sure the data was good. But with that, we have about half of them use chlorination and half of them use UV. So it was a good way to compare, how are these different treatment methods performing. For the most part, the chlorination performed better than the UV. And you can see that in both the total size of the circles, so the total sum of antibiotics, as well as in the individual sums of antibiotics. So one exception here is this Metro one, which has a little higher total sum than some of the other ones, but that also could be due to the fact that it's such a larger sewer shed that it has a more steady stream of these antibiotics because it's so much larger and it's not as susceptible to variation as some of these other ones. But overall, we found a lot of antibiotics that are used in humans: norfloxacin, ofloxacin, ciprofloxacin, doxycycline, erythromycin, azithromycin, clarithromycin, and amoxicillin. Those are all used in humans. There's also some lincomycin, this lovely, pretty pink color, which is not used in humans, but it is naturally occurring, so it's not super shocking to find. Moving on to wastewater treatment impacted places, we have Bemidji. So this is in the north of our state. And you can see that there's some similarities in some of the antibiotics that are found throughout this. We're going from upstream to downstream. But there are slight differences. So we see that also the antibiotic concentration increases from upstream to downstream. And I do want to note that with these wastewater treatment plant effluent samples, they're not actually effluent, it's where we think the effluent comes out. And we definitely could have missed exactly where that wastewater stream is, because even though we can see the wastewater treatment plant and we have a general approximate location of where that discharge pipe is, we don't know exactly where it is once we get out into the field. It's a lot more complicated than we initially thought it would be to find. We spent some time looking, trying to see if we could see it, but sometimes the pipes are underwater, and so there might be more at the bed. of the stream, and so the water might not have mixed fully with the water sample we took, which could be part of the reason why the downstream is slightly higher than where the wastewater treatment plant effluent comes out. With that, we found some sulfodimethoxine in all of these, and so that's often used in fish as well as poultry. And there is a fish hatchery kind of up here off to the side of the lake, so that could be a potential source, as well as they do stock Lake Bemidji with fish about every 2 years, so that maybe there's some excess sulfodimethoxine from those fish or from the water that they are transferred from. Yes, sorry. But yeah, again, we found, some of the antibiotics that are used in humans: ofloxacin, azithromycin, clarithromycin. There's also tetracycline, which is used in humans and cattle, but it's also natural. And the erythromycin is also natural. So sometimes we have these antibiotics and it's used in humans, but it's also natural, and we can't necessarily parse out the difference. Is this because there are humans here, or is this because it's naturally occurring here? Moving on to Alexandria, this is actually one of my favorite ones. So we have 3 sample points. We have Lake Ida up here, Long Prairie River, and then Lake Winona. You can see there's quite a big difference between these 3. Down here we have a wastewater treatment plant, and Lake Winona actually is on average 63% wastewater treatment plant effluent. So very high percentage. It's kind of a gross lake. It looks pretty much like a paint green. Fun lake to sample though. So we have this biggest one right here is sulfamethoxazole, and that is used in humans. The next biggest, sulfapyridine, also used in humans. And then trimethoprim, also used in humans. But there's also oxytetracycline, so that's used in cattle, and you can see there's some cattle farms around, but it's also natural. And then we also have doxycycline, which is also used in humans. So again, what we're getting so far is, mainly results that make sense. Especially these bigger concentrations, bigger proportions of antibiotics are, the human used antibiotics, and it is where we believe wastewater treatment plant effluent is coming out. Moving on to Otsego, this, so I want to note this is actual wastewater treatment plant effluent. It's not in the creek yet because we could not canoe up this creek. It was too small, which is part of the reason why it's so high compared to the other 2, because it hasn't been diluted into that creek. But we can see that there's a general, similar, what did I call it? Similar color themes throughout these 3, and especially between these 2. Again, similar, we have ciprofloxacin to outfloxacin, many other antibiotics that are used in humans, which makes sense, as well as some naturally occurring ones. The last wastewater treatment plant impacted sampling site I'll show you is in Waterville at Sakatah Lake. I like this one because it has a very different pattern here versus these other 2. Very different antibiotics found in the in the upstream versus the wastewater treatment plant and downstream. You'll notice that this has sulfodimethoxine, and that, as I mentioned previously, is used in fish. There is also a fish hatchery that's probably about here on this lake that's upstream, so that could be a potential source. Again, definitely missed where this wastewater treatment plant effluent came out. It's super, super low, but that's just a product of real-world sampling. Yeah. So now we're going to move on to agricultural impacted. So we're going to Lake Chetek in the far southwestern corner of Minnesota, and this has a very big amount of tetracycline, which tetracycline is used in cattle, and we have all of these cattle farms around. So it's neat to see, but we also, again, cannot parse out whether, that's naturally occurring, or that's because the cattle farms are there, or maybe it's because of both. We don't necessarily know. It also has a very different signature than down here. That kind of could make sense because this part is so narrow. It's more acting as a river rather than this huge lake, and it depends on what specific water stream we're getting at that one point. We take one sample for the lake, and that's all we have. We're not going back every week. We're not going throughout the day. It's one sample. It's a grab sample. Down in this main body of Lake Chetek, we have things like erythromycin, which is used in swine and is also naturally occurring. We have ciprofloxacin, which is also used in swine and chickens, and then we have erythromycin, which again is natural, so that's probably a more natural source coming. And used in swine. I might have just said that. Sorry. Going from there, we're going to go to Wilmer, and this has a variety of different agricultural animals around. We have turkey, we have cattle or beef, we have dairy, we have swine. So a lot of different factors around here. And we find tetracycline. It's naturally occurring, but it's also used in cattle. We have oxytetracycline, which is used in swine, which, there's a swine farm up there. And again, is natural. And we have a couple that are used mainly in humans. So unsure exactly those sources, but those are generally smaller portions of the overall total of antibiotics. This next one kind of highlights the complicated nature of looking at these antibiotics and trying to parse out those details, because we have a lot of beef and dairy farms around, but we find sulfadimethoxine, which is used in fish and poultry, and we find sulfapyridine, which is used in humans, and then we have erythromycin, which is naturally occurring in humans, poultry, and swine. So not necessarily, we don't really find the tetracycline that we found in some of the other ones that have beef farms nearby or dairy farms nearby. So it's complicated. We don't know what the practices by the farmers are nearby. We don't know how many cows, how many swine they have. These data are just based on permits, and so it doesn't necessarily tell us exactly how many animals are there, and it doesn't tell us, whether the farmer is really into antibiotics or not so much into antibiotics, or if all the animals were really sick that year or not. But the last one I'm gonna talk about is, we're going back to Wilmer, a different location in Wilmer. And again, we have a pretty similar signature between all three of these. And there's a lot of beef and dairy around, but also some turkeys and some swine. Again, find it similar to what we found other than the previous one I just showed you. Then you find your tetracycline, which cattle and humans and naturally occurring. We find doxycycline, which is used in humans and dogs, so that's kind of interesting. Maybe there's a lot of dogs around, or maybe not entirely positive of why that's there. And then the other big one is oxytetracycline, which again is naturally occurring, but it's also used in the poultry, swine, and cattle, which there are plenty of poultry, swine, and cattle around. So with that, I have a couple broad strokes conclusions based on that. So antibiotics, primarily used in animals, were found more frequently near animal farms. Antibiotics primarily used in humans were found more frequently in sites affected by wastewater treatment plant effluent. Tetracycline, doxycycline, oxytetracycline, and sulfadimethoxine were found most prevalently. And then as a general rule, the total quantity of antibiotics increased from the upstream point of the wastewater effluent to the downstream point. But overall, it's a complex issue. We can't put it into a nice, tiny little pretty box of, if there's one beef farm here, like we can definitely find tetracycline. It depends on so many other factors. So there's certainly, as much as we found a lot of interesting information, it certainly raises more questions. And so with that, I have some future work that I'm excited to get to. First, I'm going to process and analyze sediment samples from summer 2019. I'm about halfway through that. I ran into a little bit of hiccup with a column going bad, but starting to move again. And so I'm excited to pair that data with the water data. Also going to collect, process, and analyze additional wastewater treatment plant influent and effluent to be able to compare how those treatment processes are working and which ones are more efficient. Going to combine this is in collaboration with someone else who's going to combine the antibiotic concentration data with antibiotic resistance gene data to inform a predictive model. So this model is trying to, as I mentioned before, parse out why, or see, if we have a certain resistance gene there, what else is there that is affecting that. And this is a preliminary map of, so this is Minnesota, and this is one of the compounds, enrofloxacin, and just showing the darker the spot and the more concentrated the darker the spot, the more concentrated it is in that area. So she's doing that with all the antibiotics. It's going to happen with the resistance gene. It's using ArcGIS. I don't know that much about it, but it's going to be cool to see what she comes up with for that. And then there's a whole nother year to this project. I will be gone, but they're going to collect and analyze additional water and sediment samples for this in summer 2020 to refine the model. So with that, I'd just like to thank funding from LCCMR and the Butler and Jessen Fellowship, my advisor, Dr. Bill Arnold, as well as Mackenzie Pillsbury at the Cancer Center, and then all of my undergraduates, and I will take any questions.
That was a great job, thank you. Are there any regulatory limits on the concentrations of these antibiotics in natural waters?
Not that I know of. I don't think so.
I mean, they're all in the nanogram limit, so I guess these are low concentrations.
Yeah, for the most part. In natural waters, they are lower, especially in the United States. In other countries, specifically, there's a region in India that has a lot of antibiotic manufacturing facilities, and it's a little bit less regulated. There's super, super high concentrations in the waters. They're way higher than any I've seen in any wastewater treatment influent. So I don't think there are any regulations. I don't think there's like TMDLs or anything for these antibiotics.
So how do you determine what's good and what's bad? I mean, is it, could it be that, you have to consider other factors like the loading of those, just not the concentration, but the loading as well?
Yeah, so with what's good and what's bad, there's also, I didn't mention this, but there's effects on, well, bacterial communities from these antibiotics getting out in the environment. There's also effects on algae. It can decrease how much they photosynthesize, and it can actually decrease their growth. So it can affect other, and I don't remember those concentrations off the top of my head. It is larger than the nanogram per liter, but the main issue with these nanogram per liter concentrations is that they still can be high enough to promote this resistance and the transfer of resistance and the greater mutations for greater antibiotic resistance. So unfortunately, it's not, oh, once we get to this level, then it's okay, or it's not okay, my bad. Once we get to this level, then it's killing these algae. There's a lot of other negative effects when it's way below that level.
Okay, so I think my question is that so there would be a lower limit of the antibiotic, it may depend on the antibiotic, below which it won't have an effect. Is that correct? I mean, you were saying that even at very low levels, still you can get some that actually they may not be killed by it, but they would still generate this resistance.
So I don't know that there's necessarily like, oh, once it's 1 nanogram per liter and lower, it's okay. There certainly is, once it gets a little bit higher, it's certainly worse. Exerts more pressure, but I don't know a specific, I can't name a specific cutoff for at what point it's like, okay, this isn't a huge issue. Certainly the higher the number, the bigger the issue it is. So I think just trying to decrease those as much as we can would be great.
So that's just because this is like penicillin involved in the field, or?
Yes and no. I think it's also it's definitely, yes, an evolving field and figuring out all of these different things, but it's been studied for a bit of amount of time, and it's also the effects that it has are, it's not like, oh, who cares if like we're producing like 98% or oxygen instead of 100%. It's, well, this is leading to antibiotic resistance, which means we can't necessarily do, like, we can't treat like these infections, and we also potentially could maybe not be able to do surgeries or like C-sections, things we take for granted now require those antibiotics. And so I think because of the potential for really bad negative consequences is why there is a focus on, even if it's at a low concentration, it's still not great. Yeah.
Do you know if there are any practices to like clean up antibiotics in the environment, or like degrade antibiotics that are already out there?
That's interesting. I actually haven't run across anything. Some of the antibiotics do naturally degrade. So specifically one class, the beta-lactams, they have a 4-membered ring in them, so they pretty easily degrade. I only found penicillin G, which is part of them, once in the whole entire summer because it so readily degrades. But that'd be interesting. No, I definitely want to look into that. I haven't heard of anything. There's been focus on decreasing just getting them into the environment because that's easier. And I think right now, like wastewater treatment plants, I want to say using ozone is the most effective. I want to say that's the case, but it's not super popular in the United States yet. Yeah, Victor.
It's kind of an amateur question. I got 2 parts. The first part is that so it seems like the pig was the major contributor of the antibiotic, right?
They were second to cattle.
Second. So, so for the antibiotic that they contribute to, what kind? As if it's a little amateur because I was curious what kind of antibiotics specifically will impact the most.
Like which antibiotics are worse to get into the environment, or which
contribute by the cattle pigs?
I don't know that answer, Victor. No, it's an interesting question.
Because I was thinking maybe some specific type of antibiotic will degrade better, but some of them will be worse off. Yeah.
So like the beta-lactams, those are naturally occurring and they degrade pretty easily. So they're not widely found in the wastewater. They're not widely found in the environment as much because they degrade so easily. So maybe the answer is using, I don't want to say the answer, maybe an idea is using only naturally occurring antibiotics, but then you get stuck in when there's resistance, you can't do anything about it if you're only using naturally occurring antibiotics.
Sorry, the second question I was thinking about, I was kind of inspired by the past course I've taken because I was thinking about the runoff, thinking about the non-point sources and the point sources. So it seems like the thing you're working on right now is more on point sources. Do you have any idea about the non-point sources for them?
I would say it's about half point sources and half not. As much as you want to say, oh, here's a beef farm or here's a swine farm. I wouldn't necessarily consider that a point source because you have the swine there, but then their manure, it could be there. It could also be transported to a field and it could be applied there, and that's where the antibiotic could be running off. We're not considering every single source because we looked at a lot of different ones. Like I think we're considering some points I would consider non-point sources, in my opinion. Yeah.
What about groundwater? You're looking at all surface water sources. I was thinking about the septic system being maybe a source for antibiotics. We have septic systems all across the state. And also from these sources, agricultural sources, you have leaching groundwater.
I haven't seen anything about septic systems. I'm sure there's something out there. But as far as like manure applied to farm fields, I know I read one study that they found some in some of the in like the closer layers, but once you got farther down, the antibiotics didn't necessarily travel that far down to like where wells would be or
They get
anything.
absorbed.
Some of them, but they didn't necessarily like once they got deep enough to like where a well would be, unless it was a super shallow well, they didn't find any antibiotics.
Your data shows that the concentrations are much higher in the urban areas than they are in the rural areas. You were at like 1,700, and when you went out to the rural areas, you were around 30 in some cases. And they seem to be much lower, so that seems like it's contradictory to your statement that 70% of all the antibiotics are used in animal agriculture, where you would expect that you have a higher concentration in the rural areas because they use more.
Two things with that. I would not necessarily say that my rural versus, so this is a rural area, and this is 700-800, and this is more your urban, and it's 38, 18, 81. There was this super high one that is direct effluent though, so that's not super surprising to me. Shoot, I forgot what your question was now.
Well, it's about, comparing urban versus rural concentrations. Yeah.
So as much as antibiotics are used more in agriculture, wastewater treatment plants do concentrate them. So, you take this entire sewer shed and it's all going to this one point. And yes, it's getting treated, but it's not necessarily treating everything. Whereas with the agricultural, there's a lot more places it can go. It can be on that farm, it can be on a farm field, and it needs to be transported from that farm field to the body of water. So it's a little bit more complicated process than necessarily everything in this, however many square mile area is going to this one point.
So could you take your data and calculate what's the average concentration in a rural area for the whole state of Minnesota versus what's the concentration in urban areas?
Yeah, I can do that. I have not. I'm currently doing a bunch of statistics, so that's certainly something I could add and look into and see how that changes throughout. I haven't looked at that specific aspect. It'd be interesting.
But are there data available for prior to the actual, the pipe that discharges from wastewater treatment system discharges into the stream, we can monitor at that point. But just up the pipe, this is going out of the wastewater treatment system. They have data. If you don't have data on that, I think you collect from the stream.
Correct.
So are there data available so you know what the strength of the sources are in wastewater treatment system?
I would have to take that data myself because as much as they test for things like nitrogen and phosphorus, they don't necessarily test for this entire suite of antibiotics. And also, I would have to do that on the same day, around the same time that I'm going, because wastewater treatment plant effluent can be so varying depending on the time of the day, the season, depending on, if everyone's sick in the town that day. And so that could be something we look at for next summer, not necessarily something that we would do for these data.
All right, thanks very much.
Thank you.
You just stop the share, right?
Yeah.
Okay.